mixed substrate.
Sure.
And if we run into some more mixed substrate up here, we'll just stop this particular transect.
Copy that. meters, 130.
All right, Jan, Justin would like to come join us on the party line. That work for you?
Sure.
Come on in, Justin.
Can you guys hear me okay? Am I coming through?
Oh,
Hi.
beautiful.
You sound great. Welcome.
Thank you. Great to join everyone.
You want to introduce
Justin,
yourself,
can you
Justin?
introduce yourself?
Oh. Great minds think alike.
We're the hosts. We're the Jins and the Jans. We're the J's. Justin, Jen,
that's
and
right.
Jennifer. Jan, I should say.
I was going to joke that I already introduced myself and there's just a little bit of a latency, but I haven't yet. So, yeah, hi, my name is Justin Hull. I have the privilege of working with Jan at Greater Farallones National Marine Sanctuary for NOAA, and I work on the education and outreach team. Not usually working at this depth. This is pretty exciting. Usually I'm closer to the surface working with adults and students on school programs and public programs, really trying to get people excited about the ocean, which isn't that challenging, as our Nautilus viewers can probably appreciate. The ocean is amazing and inspiring. So we do a lot of programs to get the youth and adults get their hands on things like plankton.
I don't know if it'll reach you over there.
Science
So Justin, just the other day, I was telling a 5th grader about this upcoming exploration, and he was sure to tell me that there's a whole bunch of the ocean that has not been discovered before, and we spend a lot of time looking at the stars, but we need to spend a lot of time looking down at the bottoms of the ocean as well.
Hey,
he
Science.
says there's a lot more things to be discovered down there.
Oh,
hey,
sorry,
Jen.
Science.
I think Sam's trying to say something.
Yep, thanks. We are just about to finish this transect. We're about 10 meters off. So do you want to stop here to look around or continue on to 1E?
Let's continue on to 1E. We can pick up the pace. If we get into any kind of mixed substrate, we'll slow it down, but let's go ahead and pick up the pace.
Okay, 0.04 knots.
Here. Thank you.
Okay.
Yes.
Thank you. Bridge nav.
Sorry, Justin, we always give way to navigation, and navigation always gives way to the bridge, and we always have to give way to any kind of pilot need for communicating with navigation and the bridge and any other kind of troubleshooting. So we kind of slip in our little bits and pieces of information here and there.
Understood. I was going to join around the tether analysis, and I just thought I'd wait a few minutes.
Yeah, we kind of go quiet when the pilots have to deal with an issue, and we don't want it to become a big issue. You know, take care of a little issue before it becomes a big issue.
Okay.
So Justin, you and I, we have our office next to each other, and when you show up in the morning, before COVID, when you show up in the morning, you come in and you're always very cheery, and you say, J-Ro, and I say back to you, J-Ho. That's our greeting.
So Jan, Chad King is in the science chat, and he was asking about the anemone that we saw in that piece of China. And he said that there's a conversation going on about it on Twitter, and someone saying that it's ironstone china from the mid-1800s. So that's an interesting thing to think about. Now, it's on Twitter, so you know that's not a definite identification or anything, but it is interesting to think about, and I love that people are looking into it.
Yeah.
You know, we had
Did,
a nice
was
view
it,
of that
was
label
it-
on there.
Yeah, was it, Jen, was it you or was it Justin that was looking into that label?
I was looking it up. I thought it said ironstone, but couldn't tell for certain. And it definitely, you know, different things were coming up as far as the age of, but I didn't see any of the crowns in the imagery that I saw online that looked quite like what we were seeing. But it was a cursory, you know, web search. I don't know much about porcelain or china. So we may have found something interesting though. We were talking last night about the fact that some of this marine debris, once it arrives on the seafloor, it's going to stick around. It's not going to degrade. And it could be here for hundreds of years. Sometimes in the high sediment areas, it could get covered up and be buried. And sometimes in submarine canyons, if it's on the walls or higher up the canyon, it can move down slope as the sediments slide and move. But it's not going to degrade if it's something like, you know, that's more of a, well, plastics might break down over time, but they'll become just smaller pieces of plastic.
I'm doing a little paperwork here.
What's that in the center? Can we, looks like it might be a coral. Can we just get a little closer?
Pom-pom?
Any way to zoom on that?
Oh no,
Fluffy
you're right,
bit
it might be a pom-pom.
right under the lasers?
Coral there.
Just a snap zoom.
Mm.
Yeah, what is that?
It might be.
I think it's an anemone.
Get a zoom, Al?
Yeah, I think you're right.
Bob, can I zoom?
Yeah, go ahead. Zoom. Zoom. He's flying. Oh.
Oh, you're
You're
flying.
right, it's an anemone.
Huh?
Paul's flying from the left seat. You guys are trying to confuse me.
I handed him the box. He's officially flying. Okay.
Going in further.
Zoom out.
And ready
It's
to
not a
come out.
coral.
I've seen them
Zoom
roll
out.
across the bottom like a tumbleweed.
That's interesting.
Paul, now that you have the box, you have authorization to tell me to zoom also.
Sorry, I didn't get that. What?
I said Paul has authorization to tell me to zoom
Okay.
out too.
Yeah.
'Cause he's got the box.
If Bob says zoom in or out, I'm in agreement.
You support it? All right.
Yeah.
Oh, look at that. So Chad sent me a link, an email to the tweet about the ironstone china. They have a screen grab from the video, and then they have a screen grab of the ironstone china label. And then that's where they say it's from the mid-1800s. So very interesting.
So could they identify what it was, though? Look like a chamber pot or something.
Look like a bowl. Ironstone was quite nice china. Yeah.
That is
Really?
what I was thinking.
Well, some people
Yeah,
had nice
chamber
pots.
That's true.
It wasn't flat.
The Queen's chamber pot.
I'm just imagining
so
a
clean,
ceramicist enthusiasts everywhere being like, you hear 1800s and their mind just goes straight to chamber pot. That is
Ship humor. Are there any?
It was very clean though.
Yeah. Are there any wrecks in the area that could be related to that, or that the ironstone could be related
There is one wreck that I wish Bob Schwimmer was on. I see his name, but he hasn't been on since late last night, actually chiming in but in 2016 and 2017, Nautilus did some really beautifully fine scale mapping of this portion of the canyon, and the marine heritage people, they look at these the backscatter, the slope, and they have an eye for picking out the tiniest little variation from the data. And you know, something could be like 500 feet long, and they'll pick it out and find it for us. And so you know, one of the MHP contractors, MHP is Maritime Heritage Program, contractors, thinks that he found a shipwreck, a possible shipwreck, a bit further up in the canyon, a bit east of here. And I have not heard if that was actually confirmed. We don't have any planned dives. It's just a tiny little signal that at one point we were looking for something else and thought that that might be a little blip, something to investigate. So we put a little pin in it, and we'll, when we have the time and the funds, go and try and figure out what it really is. Your standard mapping, multi-beam, isn't always the best for finding shipwrecks or airplanes or pieces, because it's doesn't, we don't always request to have the settings kind of go deeper into the sediment. And when you're doing this in shallow water, you want one system, and in deeper water, you want a different system. So the standard mapping for substrate is kind of where we go on a typical basis. This area has a lot of sea pins in it.
Yeah. Well,
Was that Jan? You're noticing all the sea pins?
Yeah.
I don't think it's very tall. No.
Okay. Oh, because we can see the other side of it.
Well, it is got darkness behind where the line is.
Yeah.
And that other stuff is kind of noisy. So, but I just have a feeling it's not very tall.
So Sam, are you seeing a target?
Did you want to go over there or what?
No, I just don't want to get near it.
Oh, no, it's okay.
Okay.
I'm not worried about that.
Okay, copy that. Thanks. Sure, we were just looking at the sonar readout to see if there was, we needed an adjustment to our course, but we are on track.
I can sort of see some faint blue up in there.
Yeah.
Is that around where on the backscatter we see the lighter pixels?
I think it's hard to correlate.
Okay.
This is sonar that's mounted on Hercules and Argus. Bridge nav. Uh, 50 meters, 130.
So some of these sea pins, maybe not this particular species, but
you're gonna be getting into more wallish area here.
Somewhere between 40 and 50
wall-like.
years.
What do you? Can you see blue in there beyond the line?
A little bit up there.
Yeah, that's more noise though. Like right in between there.
In between here?
Yeah, right in there.
No, not much blue.
How fast are we going?
0.4. We could slow down.
Yeah, maybe. We're coming in at a little bit of a oblique angle there, not straight into it. But
Okay. So 0.2, 0.3?
you can go 0.3.
Okay.
So.
Bridge nav. 0.3 knots, please.
And Sam, can you show using your cursor on HiPAC what they're referring to when they say the blue? I know the blue is probably on sonar, but
It's like we're going up slope
where
a
that
little
might
bit.
be on
Yeah.
on HiPAC.
Yeah, that's correct. It's only visible on the sonar. I don't think we can correlate it from
You
the
stay out
backscatter
in
here on
front
the
though.
Yeah.
map. We're looking at it. It would be to this starboard side of Argus, which right now is the port side of the ship. So my cursor's there right now.
Oh, okay.
I don't think it's a wall. It's just
Yeah.
it's just a slope here. But we are going up slope now.
Yeah. So many sea pens, my goodness. What's that going by on the left side, pink?
Is that a nudibranch?
Was that the nudibranch you're talking about?
Yeah, it's a big one.
You want to hold
A
here?
little
It's a
deep
little
sea
ledge.
soul.
I think it's just a little
Ooh.
ledge.
Okay.
I don't think it's
Should I keep
not
coming
a
up?
wall.
Oh yeah, so that's the little bump. Maybe.
We just keep an eyeball. Yeah, keep driving ahead though, so we stay
Yeah.
in the front. Yeah, you gotta slow down, I guess.
Slow down the ship. Bridge nav.
0.2. So he catches up.
0.2 knots.
Oh, now it's looking wall-like. Why don't you stop?
Bridge nav. Hold position.
Keep coming up.
Coming up. I'm already up, but you can come up. And you keep driving ahead though. Stay right up in it so we don't fall off of here and not see what we're doing.
Okay, so enough of a ledge to come up on Hercules sonar, but not Argus.
Yeah,
on the
I
top
think
there.
it's okay. It's just a little ledge thing.
Yeah.
And then,
A knoll.
yeah, grassy knoll.
See, no.
Okay.
It had a lot of feather stars on it. Maybe it's a feathery knoll.
It's a lot nicer,
Yeah.
I think.
No.
You can get going again.
Okay.
Yeah.
0.2, 0.3.
0.3.
Bridge nav. 0.3 knots, please. 20 meters, 130.
You can stay
Yeah.
closer.
So for all you sea pen fans out there, earlier this morning, we got some very wonderful shots of close-ups of sea pens. Either it was late last night or very early this morning, which is why we're not stopping and zooming in right this time. And let's see, all of the video is available or will be available on YouTube. Look for nautiluslive.org on YouTube, and there will be video, and it's usually takes about 24, 48 hours. It might take a little longer in times of COVID because we're definitely working with a very reduced science team on Nautilus, a very reduced.
Jan, there's a little white circle in the middle of the screen there. Is that a sponge? I'm just thinking of that Swiss cheese sponge you were talking
that's a
about.
mushroom coral all closed in.
Okay.
Yeah, I second
I was just
what
looking for
Jan
that.
said.
Okay. Just want to make sure it wasn't that Swiss
Yeah, it looks like
cheese
one of my
sponge.
dog's toys.
What was the?
All the polyps are closed in.
Okay.
Starting
Sorry,
to
Rick,
drift
you're
this
giving
way.
Yeah.
You were giving a shout out for the YouTube channel.
Yeah. So there's the YouTube channel, and usually
There's also a
it's
rewind
2 to 4
function, right?
Yeah, it's got a nice rewind function too.
Okay. Meredith said the Swiss cheese sponge will look very purple gray. I also wanted to note that Guy Cochran, our geologist in the science chat, said that was a layered bedrock outcrop that we passed over a moment ago.
Layered bedrock. What forms a layered bedrock, Guy? What makes it layered?
Got another little ledge here.
Yeah. I think it's going to be more of the same.
Yeah.
On that.
It's showing up the same on Argus.
Guy said it's sedimentary rock. So this canyon was formed by water flowing over and cutting it as you would a canyon on land when the sea level was lower. So he said it's sand layer, mud layer, sand layer, etc.
Are you still moving? Maybe you got to hold up a second. Yeah, I'm getting off the screen. Oh, I guess.
Yeah, we're 0.2, but we can bump up.
Okay.
Lateral over and get myself back in the middle.
Yeah, it gets centered up on the screen there.
Yeah, the current picked up again.
Yeah, the wind's blowing that way.
So Jan, I'm looking in our notes about Pioneer Canyon.
Steady bias into it.
It said EV Nautilus mapped this back in 2016, 2017.
You can drive ahead again.
Yeah.
Were you out on the Nautilus at that time?
I was out on the 2016, and that was a very unusual cruise where we had a very mixed bag. The sanctuary, Farallon Sanctuary, had just expanded all the way up to Point Arena. And so we were intermittently mapping and exploring the USS Independence, another shipwreck, the fishing vessel Ituna, another vessel, the Dorothy Wintermote, and I think about 15, maybe it was 11 days. We mapped several areas, including Arena Canyon, way at the north end of the sanctuary now. And we did this area of, just a little bit south of this area of Pioneer Canyon. We also dove Farallon Escarpment, just west of the Farallon Islands, and just saw so many beautiful corals and sponges and shipwrecks and a lot of fish all around each of the shipwrecks. 2 new species of sponges off of the USS Independence. It was a really, really great experience. And in 2017, they mapped the rest of the canyon, Pioneer Canyon, as well as remapping a significant portion of this part of the canyon for some very fine scale mapping that we needed to have done.
122.
So, Jan, is this too steep vertically for transects? Like, do you prefer it to be flatter?
Oh, no. Our hard, we do look for the hard rugose substrate. It is a bit of a challenge. So what we want to be able to do is just keep either going upslope or straight. What we don't want to do is do too much downslope. Of course, you're gonna go over a pinnacle on the backside. You're gonna have to go down, but as long as that is just for a short distance, that gives us a
Like,
good characterization
Twist
of
is
that
enabled
kind of.
right now? Is it auto heading? Or
Do you
It's
wanna
in,
see how
yeah,
much
it's in
distance?
auto head there.
Yeah.
Right. So you can, if you just go to the XY page, what was the heading?
122?
122.
So you can just go, what we got sitting here? 2. There. 122. The goal.
Any chance to zoom where the lasers are right now, just to look at, I see a coral
Good.
shaped thing.
Yeah, go ahead, push in.
Just wanted to confirm if it's a Swifty or something we haven't seen yet. Yeah, I think
Let me know if you want to go
right
inside
at
it.
12 o'clock.
Again, let me know if you want to go inside her.
Good.
Yeah, I think that's Swifty, yeah.
It looks like it to me. Maybe Caitlin can chime in. I don't think Tom is on.
All right,
I
let's
don't
zoom
think
back
Gary
out.
is on. Maybe Meredith.
And Tina noted the
60.
Brazingid star in the middle there. Meredith said yes, looks like a Swiftia.
Yes, we've already collected a couple of different branched and the fan-shaped Swiftia.
So Jan,
Yes,
do you
similar
want
to what was collected off of Monterey. So where are we on
would this be an
HiPAP?
area of interest for a transect, or?
Yeah, I'm looking at HiPAP right now.
Okay.
We're about 20 meters off of the 1E waypoint.
Oh, okay.
Okay. So Sam, let's see if we can go across 1E and over, maybe do a transect along that contour, along the contour over to 1E. I guess we're not quite at that contour yet.
Okay, copy that. The contour that's right on the 1E waypoint.
Yeah. I
Gotcha.
mean,
You
yeah,
gotta
if we
hit
do
the
it like a
nip on
gentle
the-
kind of
Yeah.
turn, you don't have to be exactly straight.
Okay.
And just kind of hit that pixel, that light pixel next to the 1E.
Gotcha.
Yeah, that one. Yeah, or it could also go to this darker pixel that is just 2 pixels down and 1 to the right of 1E. So maybe swing down to about 6 o'clock.
Okay. And then, so the contour below 1E. Confirming that we want to head south of the 1E waypoint and then take that contour east.
Jan? Jan, if you're speaking, we're not hearing you.
Oh my goodness, I'm sorry. Oh.
Can you see where Samantha has the cursor right now in HiPAC?
Yes. And I,
Do you want to go in that heading?
Yeah.
Okay, copy that.
And this is slope or backscatter?
This is backscatter.
Backscatter. Okay, so we want to go towards the lighter Wilson. This is fine. This is fine. This is a nice mix.
Okay, so we'll be heading out east towards 1F.
Yes.
Copy that.
Okay. So we're right underneath the ship, right? And 1E is at the end of the ship.
That's correct.
Okay, so we'll just go past it, past the stern of the ship.
Correct, and then start
To
heading
where
east.
you have your, yeah, yes,
Great.
yes.
Okay.
Looks like we're getting some corals mixed in here.
You might want to look at that one up there.
Yeah, I was just thinking that.
At the one at 12 o'clock?
Yeah.
Any interest in that? To look at?
Let's take a quick look at that.
I'm just gonna go a little bit.
I think we're getting up in that into that Isadella territory that we had just left when we came on watch around 8 o'clock this morning. That the previous watch had a chance to move through.
Mm-hmm.
All right, Al, can we zoom in a little bit?
Do we want to stop here or just a quick zoom?
Jan, would this be something we would want to do
zoom.
more than just zoom on?
A little more.
I think just a quick zoom.
Okay.
Copy that, thanks.
Tina is saying
Drifted back.
Candelabrium Isadella question mark.
All right,
Pilots,
Al, let's come wide again.
and pilots, our new heading is 091.
Roger.
Yeah, it looks to be just a smaller colony of what we were observing earlier in the dive. There's some pretty large ones before we came on watch.
And Bob, if I'm reading this accurately, the ledge will continue to be along our starboard side.
Okay.
Is that correct?
I think so.
Yeah, I
Yeah.
mean, it's not
Okay.
very much. It's just more of the same
Yeah.
stuff, I suspect.
Okay. We're going to be traveling along a contour, so we may be continuing to see it.
Copy that. So I'm gonna come out ahead again.
Yeah, you're falling behind.
Did you already call in the 091, Sam?
That's correct. May take us a minute to get going in that direction.
Yeah.
Okay, sorry, one more time.
091.
091. So look at see the center view there? So
Yeah.
you need to go that, just lateral
Yeah.
over that a ways and ahead. Just kind of give it the beans ahead. You should be.
Copy that.
So you're coming in the view down there, so you need lateral and go that a ways.
Yeah, I guess I'm not that close to the wall on the right.
A little bit. Yeah, I think we just want to keep it in the center of the view there along here, and out in front in case it gets steep. Yes, lateral over some more.
So are we getting in position?
I'm sorry?
Are we getting in position
We are starting,
to start a transect?
we can certainly start a transect. We were heading down the contour line east towards 1F.
Okay, because we have to be able to have the lasers visible on the bottom for a transect.
Would you like to start a transect here?
No, I'm wondering if are we going in a direction where we're going to start one.
We are heading,
I thought
We just changed direction.
yeah.
Yeah.
We just changed direction to 091, headed east towards the 1F waypoint, which is about southeast from us
Okay.
at the moment.
Here.
So Guy is said the slope is up to the south.
Yes,
Okay.
I thought that's where we were heading to start a transect to go up.
I'm gonna take a peek at the tether too.
Yeah. So we want to be going more upslope. Does it seem like it was a long slope at the 090?
Okay, we can head more upslope
Okay,
then, Science?
Yes, more upslope, and I'm sorry, I can't tell what is 090. So
So
Sam, if you
our
could
original
show that on
130 was more
with your cursor.
upslopey.
Okay, we'll head farther upslope. Pilots, this will be 119.
Okay, that's sort of
Guy,
a
how does that
long
look?
slope. But
Oh, you want to go
that's
directly upslope?
I don't
Okay.
know. I don't know. I mean, I don't think she was happy with what we were doing there.
Okay.
A long slope.
Jan, we want to go directly upslope? Straight
Yes.
south of where we are currently? Okay.
Yeah, it looked like I'm sorry, I have to switch back and forth between my screens and HiPack.
Al, can you zoom in?
And so where you had your Sam, where you had your cursor on the starboard side of the stern of the boat, was a little more there was a little more contour there. Look at that. Oh, that looks like a black.
Tina's calling that a Baffy pathing. No, the data log.
Yeah.
Baffy pathing. I'm sorry, I don't know what Tina wrote. It's in a different language.
Okay, can you zoom out again, Al?
So if we
Okay.
can we hold here? I'm sorry, guys.
Sure
Can
thing.
we hold here?
Bridge nav.
Let me.
Oh,
let
we're
me-
holding here?
Let me check the.
You want a better view?
Yeah, I'm going to check the sample request
Yeah.
list.
This one was not on the sampling list from what I saw.
Okay.
I can
not
get in
finding it.
closer
I don't
here.
see it on the sampling list either.
I don't see it.
I just
Yep.
wanted to.
Okay.
Is the porch light on?
Okay.
Okay, zoom in.
So
Let me know if you want to go in tighter. Or a little wider.
I think, why don't you, yeah, zoom in more?
That's the end.
Are we good with this? Or?
Got
we're
good
not going to
captures.
collect, but the imagery is beautiful.
You want to see the base? Dang it. You zoom in more there, Al?
No, that's all I got.
That's all you got?
No more lens.
Good
All right.
captures on base.
Okay. So we good with that?
So let's hold here for a second, guys. Got some information coming in from Science Chat. So Tina, is this a different species of bathypathes that we typically see in this area? Because we have collected bathypathes from Pioneer Canyon in the recent past. Meredith, do you have any input on this? Caitlin? Okay, so Meredith Everett, who is running a very large eDNA program, is saying that she's seen the same in previous collections. And yes, Tina, I do believe it's very similar. It was collected a little bit further south. If I'm recalling it properly, but it is in the area, and there is a specimen at Cal Academy. It may have come from a little bit further north, either probably Farallon Escarpment, if not Pioneer Canyon. Okay, thank you, Nav. Thank you, Steve. I think we should move along.
Okay.
And start our transect up contour.
All right, so you'd like to head upslope or along, so upslope to the south of where the ship currently is, or along the contour to the east towards E or 1F.
Right. Oh, so put your cursor right there. Ah,
Okay, copy
right
that.
there. That's good. Yeah. Is that long enough? 120, 150 meters?
That is 30 meters
Oh, okay.
from where we currently are.
Okay.
We can lengthen that and go
I think
farther upslope.
Yeah, let's go in that direction and see what we can find.
Okay.
Where is it?
2 meters.
And while we're doing that, we will look at our next waypoint over at 1F.
Okay. Yeah.
Okay, I'll zoom out.
2 meters takes us all the way down to the bottom of the HYPACK screen, where my cursor is now.
Okay.
Is that what you'd like to move forward with?
Yes.
Okay.
Yeah. Let's do that.
All right, pilots. That'll be 153 as you're heading.
153I.
And science as a transect, do we want to be moving slightly faster? We've been going 0.2. Bob, do you feel comfortable going up to 0.3?
I think so. Yeah.
Okay.
0.3 is fine.
All right. Thank you. Bridge nav. 30 meters, 153, 0.3 knots.
And hopefully we can maintain this nice mixed substrate
153
get into hard.
heading.
Zoom in quick there, Al.
Looks like a couple of Gersemia and some soulless holothurians and a couple of anemones.
Okay, zoom out.
Steve confirming this is transect 8.
Correct. Transect 8.
And we'll call this hard Rugose for now until we see something different.
Okay, so no more zooming in and stuff, right? We're
That's correct.
transect mode.
Transect mode,
Yeah.
153 for 120 meters.
Right.
Jan, while we're on transect, if you get a chance, I'd love to hear a little more about eDNA. You mentioned Meredith's large collection program and eDNA, and I think that's really interesting.
Okay, hey, Jen,
I
can
think
you
so, 'cause video is not going to
talk
change,
about that?
right?
I'm going to do
So.
some bookkeeping here.
Okay. I'm not as intimately familiar with
What's that?
details of Meredith's study, but I can give some background on eDNA. So eDNA stands for environmental DNA. And this is when animals shed cells or mucus or other tissues into the water column that contains DNA, which is the recipe for building organisms. So we can collect water that contains these left off DNA and then analyze it to look at what animals were living in the region of where that water was collected. So it's a way of looking at the species in an area without having to collect the actual organisms. And so right now what Meredith's trying to do, and she's prompting me in the science chat, thanks Meredith, is collecting eDNA in the same area where we're seeing a diversity of corals to determine how well the eDNA that we can collect in the water is telling us what lives nearby that we can see with our eyes. All the species we see with the camera that we capture that way, is their DNA also represented in the water? And if it is, then that helps to confirm that that technique for sampling with eDNA is giving us an accurate representation of what we can see in the area. And the neat thing about eDNA is it might also help tell us what else is in the area that's harder to see with cameras, such as things that are living in the sediments or just too small for us to see with camera. So it's a really kind of, the field of eDNA is exploding right now in very lots of different applications, and it's something that the sanctuaries are very interested in because it's easier to protect things that we know are here. So the more we can get a good sense of the biodiversity of the sanctuary, the better job we can do in managing it.
That was great. Thank you so much, Jennifer, and also
welcome.
Meredith, chiming in the
Yeah.
science chat.
So, we're hoping to come along some areas like the earlier watch saw, which will have some large corals and a variety of corals. And that would be an area where we would consider capturing one of these water samples in one of the Niskins on board Hercules.
And these Niskin bottles allow us to collect a water sample from any depth. Is that kind of how it works? It can be triggered at wherever we are.
Yeah, that's right. They pull. It's so fun to watch the manipulator arm pull a loop, and that opens the bottle, allows water to flow in, and then it closes, and that water is now sealed in tight and will return to the surface on Hercules, and then will be processed in the science lab. On board, I believe the eDNA processing, at least the preliminary part of that, is done on board. We have a nice looking deep sea sole right there in the middle of that rock formation covered in feather stars. They're such beautiful fish, I have to point them out. Okay, yeah, so Meredith's saying that eDNA is filtered and then preserved on board, and then it'll be run through equipment on shore that will help to sequence it, and then you compare the sequences that you have in your sample to the known sequences in a database to help identify the species that are in the samples. Now you can imagine if you don't have a species yet in the database, you will not be able to detect it in your sample. So there's still ongoing work to actually make sure we have all DNA sequence from all the different species that we're seeing. And I believe some of these voucher specimens we're collecting will be helping with that, the DNA library effort as well.
I imagine when that bottle arrives of your preserved specimen, it must be pretty exciting to be like, now I get to analyze it, see what's there, and see what I think is there. I don't know if Meredith, if you have any thoughts about that, about whether that's sort of like a big red letter day of gifted specimen from the deep that is challenging to collect. I imagine that's pretty exciting.
Well, it's always fun to look at the first pass of sequence as the data comes in, is what Meredith said. Well, when I've had the opportunity to be on board Nautilus, I think it's so exciting when the samples come on board and we get to offload them and see these animals that I've only seen on video in person. And in fact, when we collect sponges last year, our sponge expert, Amanda, she even said that sponges smell different. And one of the things that we were doing was smelling the sponges. And they really had some interesting aromas.
What type of aromas are we talking about?
Garlic. One was kind of garlicky. Another one she said she thought smelled like mowed grass. And I never would have thought to smell them.
What did they taste like?
Not
didn't
like
go that
maple.
far. All of them were more pleasant than those of us who decided to smell the whale bone we brought up last year from the whale fall. That was something. Only need to do that once.
When you said that it smelled like fresh mowed grass, I'm picturing an aquatic animal passing a lawn that has just been mowed and being like, that smells like sponge.
And not just any sponge, it was a specific species. I don't remember which one. But at some point, Amanda Kahn might pop into the science chat, and she's pretty fun to talk with about sponges. She got everyone excited about them during her watches.
Yeah, she found, we found what was turned out to be a new species of sponge. It's related to one of the carnivorous sponges, but it's flat. And last year when we were up off of Wallala, Sonoma County, we found these flat, leafy carnivorous sponges, and the plankton was just blooming. The zooplankton, the krill, and it was just a beautiful sight. The sponges were capturing with their little barbs krill, and you could see some of the krill kind of embedded inside the sponge itself. They're flat, like a palm sponge. And Amanda and Henry Weiswig, who passed away recently, and another fellow from Mbari are writing up this new species of sponge.
So science, we
That's
just
pretty
passed the
interesting. I wish I'd seen that.
halfway point of our 120 meter transect. When we reach the end here, do we want to continue east or continue upslope? Or sorry, continue upslope or start heading east towards 1F? I can zoom out here on
Okay,
HiPAC.
so let's take a look at HiPAC. And if you can get
Zooming out here for 1F.
zoom out a bit, and let's see point 1F is. That looks like a pretty interesting steeply sloped area between 1E and 1F.
Yep, there's 1F on the far right side now. And from the end of our transect, it will be about 621 meters away.
Seems like
So
we just
if we get to
transitioned
another
into soft.
stopping point somewhere,
Yeah.
I think I want to take a gander of the tether again just to make sure we're
Okay.
A-okay.
Yep.
Okay.
Science, can we just stop at the end of this transect then? And
We just need a second to just kind
Yeah.
of take a gander, see what's going on,
okay, perfect.
just to make sure.
Okay. So going back then to contours, do we want to head directly east from the end of this transect or follow a contour line to get to 1F?
No, I think what we want to do is try and zigzag our way. We have plenty of time to be able to go upslope a little bit and then go back down and go back upslope and go back down across this. You see where the contour lines are really, really narrow, about 2 boat lengths from the stern. Yeah, that whole area. What do you think about that, Guy? Because this whole ridge is pretty steep, and that's kind of what we want to capture as we go across. So if there's a way of describing that.
So Jan, are you saying we would, when we finish the transect, we might follow the contour a little bit east and then go upslope where the contours are tightest?
Let me look at HiPAC again.
Guys chiming in. He says, or crab east facing south.
Yes. Let's see if we can do that.
Okay.
Guy, can you see Samantha's cursor? It's a little white cross on HiPAC. It's right on a contour line. And if you can direct
Yes.
which contour line you want to crab across, is it like 2 above that or 3 above that?
Yep, I just dropped a target there on that contour line. And for the pilots, we've got 1-2 meters to the end of this transect, and then we can pause for you.